Got you. Hello, everybody. I hope you guys are doing great. So my name is AJ. I am a intern here at Archbold. I'm a Vaughn-Jordan Ecology program intern here at Archbold. So to give you some background about me, I'm in my post-baccalaureate internship. I graduated from Virginia Tech in May of 2020, and I'm going into my master's program in this fall. And I've really enjoyed my time here at Archbold. I really enjoy the people here. I love what I do, which I'll be talking about in a little bit, but I work in the plant lab, as they said, multiple times. And we typically have 2 to 3 interns, and we come from everywhere. We all have different backgrounds. To give you a little idea, some people come from entomology backgrounds. I came from a wetland biogeochemistry background, so very diverse. Can you click next for me, Margaret, so I can talk about what we do in the plant lab? So I thought of the top 4 things that we do. And I think the most important things, we do a lot of field work, demography, which is just looking at what's there, what's alive, getting measurements. We take clippings, we do introductions of plants. And with field work, that means we do a lot of data collection and analysis. We have a herbarium, which I'll be showing you later on. We do a lot of upkeep of that. And then every intern has an intern independent project. So right now, I'm kind of still going through mine, but hopefully the next time I talk to you guys, I'll be telling you guys what I learned in my internship project. And then this next slide is some pictures of our field days, which can be really long, but they're so amazing. I'm going to start with one of my favorite projects, which is Florida Ziphysis. Can everybody say Ziphysis for me? It's one of my favorite words now. And we did demography and sampling the whole month of January. It was my first project here at Archbold. And Ziphysis is just a really interesting plant. It was, endangered for a really long time. They thought it was extinct until 1987. It was rediscovered. And then in 2001, 8 more populations were discovered. And then that's when a lot of places like Archbold and some universities took on projects to then keep Ziphysis alive. So since those projects have started, they've planted 600 plotted plants and about 6,000 seeds. That's a lot. So just to keep you in mind of how important Ziphysis is and how important it is to keep rare endangered species of plants in this area in Lake Wales Ridge alive. In that bottom left, you can see we're doing some collections of Diceranda funiculata. And that's a rare mint species here in Florida. It smells so good. And then on the bottom right, that is us out there. It was really cold that morning, contrary to believe. It gets kind of cold here in Florida. And we were doing Florida rosemary demography. So as you can see to the far right, that is our RA, Lexi, with her retractable measuring Florida rosemary, which can get really big. And we were looking at the sex, whether it was male or female or not. And yes, it was a great project. I enjoyed that so much. So those are my favorite 3 projects so far that we've done. But like I said, I'll be here for a while longer. We have so many more things to do. So hopefully I can tell you guys about that later as well. Okay, so we're going to do a quick tour. I'm going to start by saying hello to our RA, Lexi, and our other intern, Haley. So I'm just going to show an old intern project. I hope you guys can see it. It's a lichen project. I don't know much about it, but it's a different species of it. Really amazing. And then we're going to go downstairs because I have some really cool tools for you guys to see. So what we do in the field is mostly just maybe tagging plants, checking. When we do demography, we're checking to see, the plants that we may have tagged 3 years ago, are they still there? How tall are they? Have they gotten any wider? Are they reproductive, actively reproductive? So I'm going to show you guys. Let me turn around my camera. All right, this right here is Lucky. This is our metal detector, which detects our tags and our flags so that if we ever lose a plant or we can't find one and we need to use our GPS, we can use Lucky to just scan the area and hopefully we catch something. These are our 2 different types of GPSs. This is the Garmin, a little bit smaller. And then this is the Trimble, super expensive, nice and heavy, but this can get you pretty much to anywhere. Yesterday we did Diceranda Christmanii, and this is like one of the maps from there, and this is just like a data sheet. These are the tags. We have so many tags because we have so many plants. And then sometimes our plants are really tiny and we can't use tags and we can't use flags. So we have to use little toothpicks, and they all have these different shapes. So we have spades, we have hearts, we have diamonds, and they're all in different colors just to identify them even more. And then one of my favorite parts of this is we have a herbarium here. So I'm going to show you guys how we mount our plants. After we use this really cool adhesive glue, we then, kind of like paint it on to these plants, and then we mount them. So these mounts are like little paperweights, and we use them to hold the plant down so they stick a little bit better. And then when it's all done, it looks something like this, really pretty. And now I'm going to show you guys our huge herbarium. So many awesome plants. There you go. So just imagine every little one of these folders has maybe one, maybe two plants in them. And it's 3 containers of them. So many of them that had to be hand pressed, hand dried, hand, glued, and then mounted, and then put into these in a certain order. To just give you some ideas of like what goes on in here. That's just a little bit of it. Oh, and one of my favorite parts of this, for obvious reasons, is this huge map of Archbold, which I'm going to end with because I don't know if you guys noticed, but Archbold is pretty big. And I think that giving this perspective of it, of how really big it is and how many lanes we have and how many areas we have, it's really beautiful. So this is another view of our lab.